Well, thank you. Okay, it's time to get going, so if everybody could have a seat and get ready, get out what you need, the whole 9 yards. A couple of items. Number one, I just want to tell everybody how much I appreciate you being here today, those of you who are. And those of you who cannot, that is completely and totally fine. Safety first, foremost, and always. Number two, I apologize for the lack of heat in the building. I'm up here suffering right alongside the rest of you. If you need to step out and warm up, feel free. You don't have to ask permission, okay? I am definitely recording the lecture this morning. So a couple of you mentioned you have difficulty hearing, so I'm trying to speak up a little bit more and sit close. I will try to tame my predilection for wandering about the classroom today, which is hard because it's even colder, but we'll persevere. Last item, make sure you all are aware that we are going to have an exam 1 week from today. I believe it's scheduled. Okay? We will have that whether or not we have another bout of weather. Okay? Because I try my best to make sure that the test dates are more or less engraved in stone, so if you have an issue, you know that you can work around that date. If we end up not having it, I will post a study guide for you, and I will make the exam directly from that study guide. Is everybody clear on that? Does that sound okay to everyone? And what we're going to do is I'm going to shoot my best to get through chapter 4, so it should be over chapters 1, 2, 3, and 4. Everybody clear? Okay. So I believe the last thing I discussed was mitosis. Is that correct?
Yes.
So remember, whether or not you're looking at mitosis or the cell cycle, it's essentially the same thing. It just depends on what you focus on. I'm a geneticist. I'm far more interested in the DNA and the chromosomes, so I know a lot more about mitosis. Someone who is a cytologist or a cell biologist, they're going to be more interested in the whole thing, so they're going to focus on the cell cycle, right? You need to know the steps of both. Either of those would be a great exam question. Question number 1, draw and label the steps of the cell cycle. It's up here on the board. You've seen it, right? Another question would be list and describe the steps of mitosis. Now I might ask you to list and describe the steps of the cell cycle. It's just different ways of phrasing it. But that also means you get different ways of answering it. So if you choose to draw it, that is completely fine. If you prefer writing it, that's also fine, okay? So is everybody clear on that? The whole point is for you to show me that you understand these two processes. Okay, so what happens when you reach the end of the line? And by that, I mean cell death. The technical term for this is senescence. Senescence.
Can you spell that?
You would ask me to do this.
Sorry.
I was about to ask.
Senescence. Senescence is essentially, for lack of a better term, when the cell wears out. Old age. That's not really a good term, but hey, this is a first year class. We'll put it in simple terms. Everybody okay with that? And every cell has the potential to eventually undergo senescence. No, pass it this way. If you pass it straight back, it'll never get to everybody. What contributes to the wear and tear? Variety of things. Consider a red blood cell. Red blood cell lasts about 90 days on average. Consider the path that red blood cell takes throughout your body. It's going through the heart, which is beating sometimes faster than once a second, right? Think of the blood pressure, 120/80, that's pretty high. It's forcing a liquid through a very narrow set of tubes, isn't it? Think of how fast it moves through. You probably recycled your entire blood supply in less than 5 minutes. It's getting banged around pretty good, isn't it? Against the walls of the blood vessels, against other cells, against proteins, and eventually it just gets dinged up. It's just like an old beater car, right? I'd be willing to bet most of you are probably not driving a brand new car because at this point in your life, you probably don't have a lot of liquid assets and probably can't afford some. But you probably got yourself an older car, may have a ding or two on it. That's just the nature of the beast, right? Cells are the same way. The other contributor, or the second contributor, I should say, would be environmental damage. If you go for a walk on a sunny day, you're getting damage to your skin, aren't you? Ultraviolet radiation. It doesn't matter if you're only out there 5 minutes. That's 5 minutes of radiation exposure. Now, cumulatively, that's not going to be dangerous, but you see where I'm going. Wear and tear. Do you get exposed to chemicals, things in your food, things in the environment? You ever gotten behind a bus, it takes off, and you get that big cloud of toxic black smoke comes in, and you're hacking and wheezing? You just got a lung full of hydrocarbons, didn't you? See where I'm going with this? The third is damage due to metabolism. Believe it or not, the oxygen that you breathe that gives you access to energy is also probably what ultimately kills you. Why? Because oxygen is one of the most electronegative elements. You don't have to get that particular term, I'm just telling you what it is. Is oxygen very reactive? Yes, extremely so. Electronegativity means that it wants electrons. It will get them from whatever is available. Does that include proteins in your cells? Yes. Does that include DNA? Yes. Do you see where I'm going with this? Eventually, you just simply accumulate so much damage that the cell is no longer viable. And at that point, it will generally undergo apoptosis. I did talk about apoptosis.
Cellular suicide?
Yeah, cellular suicide. It sounds graphic, it sounds bad, but the simple fact of the matter is your body uses up cells all day long. You are losing cells just by sitting in your chairs. You move, you shift, you wiggle. Your clothes rub against you, don't they? They rub against your skin. A few cells slough off. When we start talking about tissues in the next chapter or two, you'll see what I mean by that. It's normal, it's part of life. That's one of the reasons we change as we grow and get old. When you hear somebody dying of, quote, old age, generally what it means is you've simply just accumulated enough cellular damage over a lifetime of living that there's not enough new cells to make a difference. Everybody understand that? You don't die of, quote, old age. You'll have some kind of organ failure, but it's typically due to the cells in the organ just not being functional. Okay, any other questions about aging? Did I talk about the Hayflick limit?
Yes.
Telomerase, cancers, embryos? Good. I think we've covered that enough. So now we're going to move on to chapter 4 and start talking about cellular metabolism. Metabolism can be broken down into two separate paths. The first is called anabolism, or anabolic. You may have heard of anabolic steroids. This is building up molecules. You go from small to large. Look at any of the walls in this classroom and you can tell that they're made up of individual blocks, right? Each block is small, but if you use mortar between the blocks and you use energy to lift them up and set them into place, you can create a wall out of the smaller blocks. Everybody see how this goes? Notice though I said you have to lift them up. This requires energy. This requires energy. Every cellular process requires energy. So anabolism or the anabolic pathways are building up. If you look at the cell cycle, you see that growth is an anabolic process, isn't it? You're putting energy into the system. You are making big molecules from small ones. Protein synthesis is a great example of this. DNA replication. Everybody got the idea? Contrast that with catabolism, or the catabolic pathways. As you might imagine, catabolism is breaking down rather than building up. You are starting with a big molecule and you are breaking it down completely. So look again at that wall. If we started swinging a wrecking ball at it, we would knock it down and it would be broken up into chunks. You'd be left with a pile of rubble. Does that require energy? It does to initiate it. But the key thing I want you to take away from this is anabolism requires energy. Catabolism releases energy. So if you need a new protein, that's an anabolic process. It costs you energy. Am I going too fast? Catabolism means you are going to break a big molecule down into smaller molecules, and in the process, you are releasing the energy that is found in those chemical bonds. Instead of having to put energy into the system, we are getting energy out. this is not 100% effective. It is not completely efficient. A large, yes ma'am.
Sorry, could you repeat the part about releasing energy found in the bonds?
It releases the chemical energy found in the chemical bonds. So if we split H2O, which is water, if we split that into hydrogen and oxygen, we're releasing energy, aren't we? I know, it's cold. I have a spare jacket if anybody needs it or wants it, but you'll have to run to my car and get it, and I will warn you, it is UT neon orange. I know most of you would refuse to wear that, seeing it as enemy colors. Anyway, back to metabolism itself is the sum total of all these reactions. It is the sum total of all of the reactions. It may seem like I'm being a bit pedantic or overly picky in my word choices here, but remember on day one we talked about science, we talked about hypothesis, we talked about a theory, and then a theory is actually a well-supported hypothesis. It doesn't imply that there's uncertainty, it implies this is as certain as we know it, right? Well, everyday lingo, people say, oh, I've got a fast metabolism, meaning I burn a lot of energy, burn a lot of calories. Well, no, what they're really saying is I have a fast catabolism. Okay? In terms of physiology, metabolism means both pathways. Make sure you know that, understand that, and have it. What type of energy are we looking for here? Does anybody know?
ATP.
ATP. Good job. Somebody's been doing the reading.
I have a question.
Yes, sir.
You said it's not 100% effective at a large, and then you stopped.
You lose a large amount as heat. Yeah, that's when Ava actually made me do my job by asking the question and totally ruined because I was that close to the punchline. It's so cold in here talking about heat. Way to go, Ava. Make me do my job. Y'all know I much prefer it when you ask questions. And thank you for catching it, Jason. So everybody understand you lose a lot of it as heat. Heat is a waste product, generally. You can overheat, can't you? I know it's hard to imagine in this classroom on this day, but I assure you it is possible. Now, make no mistake, in some cases you want heat, but when I say a lot of it is lost, I mean that. It's not a very efficient process. If you start tracing energy through a food chain or a food web, you end up losing about 90% of it at each level. So please understand, when I say it's not very efficient, I mean it's not very efficient. But the end goal here is ATP, adenosine triphosphate. Tri means it's got 3 phosphate groups added. Those bonds between the phosphate groups are considered to be high energy bonds. When you break them, you release a fairly large amount of energy, at least on the molecular level. When you build them, you take a DP, which is adenosine diphosphate, meaning it has 2, and you add another phosphate group to it. That requires a fair amount of energy. Yes, ma'am?
Could you repeat that?
That requires a fair amount of energy.
I meant the part before that, about what it takes to build
Where what?
What if he's, oh, never mind, she got it.
Okay, y'all need to tell me to slow down immediately. I don't think it rude if you interrupt me, okay? Problem is, I've been teaching this so long, I know it so well, sometimes I get on a groove and I just get rolling. Back in the old days, before you were born, society had this thing called cash. I'm pretty sure none of you use it, but you may have seen it. Your grandparents, maybe even your parents, had it at one time. Might have even held a dollar bill in your hands. Watch an old movie, you see piles of it, right? What is good about cash is that it can be used for anything. If I wanted to go over to Starbucks and get myself a cup of coffee, I could pay for it in cash, couldn't I? I need to put some gas in my car on the way home tonight, I can pull over to the gas station, I can pull out a $20 bill and pay for it with cash, couldn't I? If I needed to pay my income tax, hypothetically, I could use cash for it, couldn't I? I could stop at the city hall on the way home tonight and pay my water bill with cash. You see where I'm going? It's useful for pretty much any transaction. ATP is like cash. It can be used by the cell for pretty much any cellular process that requires energy. The cell can use ATP for any cellular process that requires energy. You want to make a new protein? ATP. You want to bring in a molecule from the outside using active transport? ATP. You want to make a copy of your DNA? ATP. When you lose ATP, you lose life. It is essential, it is required, and that is really truly what most people mean when they talk about metabolism. Getting ATP. Now let me make sure I'm not getting too far afield here. I tend to get rolling. Metabolism is not possible without enzymes. Metabolism does not occur without enzymes. Enzymes are proteins that act as catalysts. What does that mean? Well, if you look in chemistry, they'll tell you that a catalyst is something that speeds up a reaction, right? But in humans, people, and living things, it has a rather more specific definition. It lowers the energy of activation. It lowers E sub A, the energy of activation. What does that mean? Well, we just said everything requires energy, right? We might get more energy out of it than we pay into it, but we still have to put some in at the start. Is everybody with me? You want an example?
Yeah.
All right, take a look at the attendance sheet here. Sheet of paper, right? If I wanted to burn this, Is it going to burst into flame simply because I am holding it in my hands?
No.
Can I make it burst into flame simply by staring at it? No, I'm not like the illegal alien superhero who wears the red and blue long johns and shoots laser beams out of his eyes, right? What would I have to do? Somebody tell me.
You would have to apply heat or energy, put energy into burning.
Put energy into it. Thank you. Apply heat. I'd need to strike a match and hold that, wouldn't I? I'd need to get a cigarette lighter and hold that, wouldn't I? You see where I'm going with this? That is the energy of activation. It is the energy that you put into the system to make the reaction occur. It is the energy that you put into the system to cause the chemical reaction to proceed or to occur. Now,
Word.
has anybody ever made caramel at home? A couple of you. You have to get an open pan and heat it, and you put sugar in it. So you're literally heating the sugar, right? It gets nice and brown and gooey. We like the taste of it because it's primarily sugar. It's sweet, right? You are literally burning sugar on your stovetop. But I promise you, none of you are going to stick your finger down in that saucepan and get some and lick it off, right? Because if you do, the temperatures on that are going to be so high, it's going to burn your finger and your tongue. It will damage the tissue, right? I bring this up because so often people say they're burning sugar in their cells. Isn't that what you've been taught? So how on earth can your cells burn sugar at body temperature, which is so much lower than what's actually going on the stove where you're really burning sugar? The answer are enzymes. Enzymes lower that energy of activation to essentially body temperature. They do this by catalyzing the reaction. And the thing they work on is called the substrate. The substrate. S-U-B-S-T-R-A-T-E. The substrate. Substrates and enzymes are specific. And I mean specific.
Can you repeat how to spell that?
Substrate,
Yeah.
sub, S-U-B, straight, S-T-R-A-T-E. And for those of you following along in your books, we're on section 4.2 under enzyme action. Very first day, I remember, a lot of you were sitting out there in the hall when I got here. Remember that? You had to wait for me to unlock the door. Now, on my key ring, there's a specific key that says C111, Cooper 111. That's the designation of this classroom, isn't it? Right next to it, there is a different key labeled C114. That's for 114, which is the lab. Now, if you look at these, they're very similar. However, they are still different because they have different patterns, right? I may very well be able to get the key for C114 into the lock on this door, but will I be able to turn it? See what I mean when I say specificity? This is actually called the lock and key model. The lock and key model. Every enzyme is like a lock. Every substrate is like a key. It fits, and it fits perfectly. This is one of the reasons why enzymes are so crucial for life. Because if you change the structure of the enzyme, even just a little bit, it will no longer function, or if it does, it will be at a reduced rate. A lot of recessive mutations actually reduce enzyme functions. Now, like I said, there's a whole field dedicated to that, genetics. As much as I'd love to get into it, I can't, because we'd never get off it. So everybody understand how enzymes work? Generally, what limits the rate of the reaction is likely to be either the amount of substrate or the amount of enzyme. Enzymes are generally named with a suffix ASE. They have as a suffix ASE. Lipase, LIPASE, that breaks down a lipid. Hydrolase, ASE, adds water. Amylose is starch, amylase breaks it down. You see where I'm going with this. So anytime you see a word ending in ASE, it's probably going to be referring to a specific enzyme. The substrate binds to the enzyme at what we call the active site. The active site. Enzymes are often components of what we call a metabolic pathway. A metabolic pathway. So let's say we start with a big molecule like sugar. We break it down completely to water and carbon dioxide. Those are small molecules, right? Do we do that in one step? Do we go straight? No. That process requires multiple steps. So rather than going straight from A to B, we go from A to B to C to D to E. You see how this works? And in the human body, there are hundreds of metabolic pathways. When I moved, I lost my poster. I wish I could find it because I used to bring that in. It was a visual representation of the biochemical pathways in a human cell. And it had the Krebs cycle at the center, and it had every possible way it could feed in every step. And it looked essentially like a bowl or a plate of spaghetti because all the lines led everywhere. These things are interconnected, They have multiple switching points, they have multiple feed points, you get an idea? So we may have 1, 2, 3 lead into step C. It doesn't have to be just A to B. We could start with number 1, whatever number 1 is. You see how it goes? Don't let me put you to sleep. I don't feel bad. I hate biochemistry. Of all the things in the field of biology, this is the thing I like the least. These pathways are regulated by a single step. It's called the regulatory enzyme, or the regulatory reaction. The regulatory enzyme is the slowest, typically. Case in point would be if we all wanted to get into a race. Let's say we started out here and we had a team, we would go around the laps. If I let you go as individuals, we would see quite a range of times, wouldn't we? Somebody in here would be the fastest. Who's probably going to be the slowest, do you think? You could say it, I'm setting myself up here. Y'all know I have back issues, sometimes use cane, right? If I had to race a bunch of 18, 19, 20-year-olds in laps around the campus, I think it's kind of obvious who the slow guy is. But if I made us go as a group, I set the pace. See where I'm going with this? So everybody understand how these work? The regulation of metabolic pathways is very complex. It is very complex. We're not going to get hung up on that. I just want you to have an idea how it works. Everybody with me? Now, sometimes these enzymes have to have helper molecules, helpers. If it's not a protein, it is called a cofactor. A cofactor. If it's another organic molecule, it can be called a coenzyme. A coenzyme. A lot of coenzymes are vitamins. Now you know why they tell you to eat your vitamins. Or vitamins if you prefer the British pronunciation. I can't tell if Haley's smiling or not. Question? Just trying to warm up. A jacket is a good thing on a cold day. Finally, there is something called denaturation. Denaturation. If you denature an enzyme, you cause it to lose its three-dimensional structure. You cause it to lose its three-dimensional structure. You're not breaking it down into amino acids. You're just breaking the three-dimensional structure. The best example I can give of that would be an egg white. Has anybody here ever cracked an egg? You don't get a solid white substance coming out of it, do you? You get a very runny liquid that's generally translucent and may have a pale brownish or yellow color, right? It's only when that liquid hits the hot grease in your frying pan, or it's heated for several minutes in hot boiling water, does it become a solid white mass? Y'all seen that? You ever fried an egg? That is because the protein, which is called albumin, has been denatured. When it is in its normal form, albumin is actually water soluble. You have albumins in your bloodstream right now. But when you heat it, you denature it. You destroy the three-dimensional structure, and instead of being water soluble, it is now that lumpy white substance that is a solid. And generally, denaturation cannot be undone. You can't put the white back into liquid form and stick it back in the egg, can you? Does everybody see how this works? Any questions? Generally, the things that will denature a protein, number one is heat, high heat. And number two would be acids or bases, an acid or base. Is everybody with me? The advantage and disadvantage, I think you see. It's when we talked about buffers, we talked about acids and bases, right? You see why it's important to have buffers in your bloodstream. You also have buffers in the cytoplasm of your cell. Everybody take about 30 seconds or a minute and just, I would suggest standing up and stretching or maybe moving a little bit because it's a little cold. Y'all are starting to get just a little bit of that 1,000-yard stare. Moving may help. Don't worry, I'm not going to make you march in circles around the hallway. But if you want to come up by the heater and warm up for a minute, I'm completely okay with that. Is this helping a little? A little? Feel a little better? Well, there goes me making any effort to accommodate you. Okay. So what we're going to talk about next are chemical reactions, the energy, the actual energy involved. And basically, this is a series of what we call redox reactions, R-E-D-O-X, redox. Redox is a shortened form of reduction and oxidation. Reduction and oxidation. Checking to see if it's on high. It is, unfortunately. Now, what is reduction and what is oxidation? These reactions always occur in pairs. If you have a reduction, there has to be a corresponding oxidation. If you have a reduction, there has to be a corresponding oxidation. Y'all want to use the little cheat my organic professor taught me? Organic chem professor taught me. Oil ring, O-I-L-R-I-G. Oxidation is loss Reduction is gain. Oxidation is loss, reduction is gain. Loss or gain of what? Electrons. Everybody with me? So in other words, if Jake knocks me on the head, he takes $2 out of my pocket, I've been oxidized. He's been reduced. Thank you for letting me use you, Mr. President. Helps y'all remember it though, baby. Essentially, what it amounts to is one molecule is going to lose the electrons and they're going to go to another. And you're going to see that in humans, essentially, respiration is just passing along those electrons. You start with getting them from a bigger molecule, let's say sugar, and you pass them down eventually until the ultimate recipient is going to be oxygen. Everybody cool with that? And again, I am glossing this over at a very, very simplified level because this isn't a biochemistry class. It's not even a cell biology class. And be grateful. Because in biochem, you actually have to know each and every reaction, each and every enzyme, the structure, and how long it takes.
What's respiration again?
It's just basically a series of redox reactions. You're passing electrons along. That help? And we've already mentioned ATP is the goal here. So, respiration as a whole. There are different ways you can break this down. Your book simplifies it. So we're going to get the three as listed by your book. Does that sound okay? The first step is called glycolysis. 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 Now, as you learn the language of science, you know that anytime you see GLYC, you're going to be referring to a sugar, right? Lysis, Lysis, always means splitting. You're breaking something apart. In this case, you're splitting the sugar molecule. This occurs in the cytoplasm of the cell. Once this part is done, you enter what's called the citric acid cycle. And I promise you, I will make an error at some point in time and call it the Krebs cycle because that's who it's named after. And that's how I learned it. And as Axel will tell you, it's absolutely the worst thing to learn in the whole field.
You have to memorize it. It's like 9 reactions.
Yep. It's a real pain, wasn't it?
Yeah.
Don't worry, we don't have to memorize it for this
9
class.
reactions of complex names. Gotta remember all that.
And no, even with 40 years experience in the field, I couldn't tell you the name of every single reaction. I have to look it up myself. There's a reason I'm not a biochemist. And a lot of it has to do with Herr Krebs and his darn cycle. So everybody with me? Krebs cycle takes place in the mitochondria. Takes place in the mitochondria. Is that a shrug or a twitch?
A cold chill.
Huh?
I got cold chills.
Oh, you think it's bad to hear? Wait until we get in the lab. It's cold, I'll agree. And you're talking to somebody who likes it cold. My students used to refer to my office as the meat locker because I kept it so cold. But this is too cold even for me.
What's the temperature?
So, if you look at the mitochondria, remember it was kind of shaped like a bean, but it was hollow, wasn't it? You remember the inside of it looked like it was wrinkled? That's the inner membrane. It's that way with the wrinkles to increase the surface area. The Krebs cycle takes place in that hollow space right in the middle, okay? Everybody with me? And then we move into the last phase, which is called electron transport. Electron transport. The electron transport takes place in proteins that are stuck in that inner membrane. It takes place in proteins that are stuck in or part of that inner membrane. Does anybody here ever play with a slinky as a kid? Y'all know what a slinky is, that coiled spring? You may have seen the commercial where they start it at the top of steps and flip it and it just kind of walks itself down. Electron transport is kind of like that slinky going down the steps. You lose energy at each step. What's actually going on at each step, they're using that energy to set up a proton gradient. A proton gradient. Now remember, you've got the outer membrane of the mitochondria, you've got the inner membrane, right? So that means you've got a space between them, doesn't it? And what you're doing through electron transport, you're bringing protons from the center space up to that space between the two membranes. Everybody see that? That's a high concentration, isn't it? They're going to want to go back into the center because you pulled them all out of the center. Everybody agree? When they pass through a special enzyme that's located on that inner membrane, they actually cause that enzyme to turn like a wheel, and it smacks an ADP and a phosphate together to make ATP. You're done.
The protons just go through like the wall of the inner membrane, or you're saying they just go through like one little section?
They go through the inner membrane.
Like
Are
through like
you
all
talking about
the
when they're
way?
coming in?
When they're going out, you said it's going out from the inner membrane to the like the outer space?
To the intermembrane space, which is the space between the outer and inner membrane. Okay? We're moving them up. Imagine I'm lobbing beanbags or balls up into the ceiling. Okay, imagine the ceiling allows them through. Everybody with me? But we've sloped the ceiling to where it's kind of like a little funnel. So when the balls come back out, they cause a wheel to turn. That help?
Yeah, that makes sense.
And don't feel bad, it's not easy to visualize. It's probably more detail than you want, maybe even more detail than you need. But your book only just says electron transport, and every time I taught it, people would always say, but how do we get ATP out of that? Well, you really need to add step 4, which your book doesn't. Don't ask me why. So everybody got that? If you want to see a summary of this, take a look at figure 4.11. It's on page 60 in the electronic version. And I believe page 133 in the hard copy.
It's 134.
134, excuse me. Thank you for the correction, Axel.
Wait, where is the electron transport chain? Where is it located then?
The inner membrane of the mitochondrion. Don't worry, I am not going to make you know this at the level of a biochemist, okay? What did you end up taking? About a half a page to describe all that, I think? Yes, no, maybe? Nod, drill, or otherwise indicate consciousness. I know we're cold, folks. I am too. I'm here. You are here. Let's make the most of it. Boy, what a day to be teaching cellular respiration. It's terrible. Now, the thing I want you to understand here is this process requires oxygen. This process is aerobic. A-E-R-O-B-I-C, aerobic. It requires oxygen. How much oxygen? Well, here's a simple rule for you. The rule of 3. How many weeks can you go without food? 3. How many days can you go without water? 3. How many minutes can you go without air?
3.
And having been dead for 2 minutes, you see I was pushing that. 0 of 5 stars. Do not recommend. Do not wish to repeat this experience. So does everybody at least understand the basics of cellular respiration? If you do, give me a thumbs up, and I want to see everybody give me a thumbs up before we move on. Okay. If you've just got the basics down, that's all we need. The point I want everybody to understand, this process has to be continuous. It requires oxygen, and you simply cannot survive without it. Hydrogen cyanide, that's the gas they used to use in the gas chambers for executions. It blocks the electron transport chain, so no more ATP gets made. Anybody here ever run a long distance or really had a hard workout? How do you feel the next day?
Sore.
Sore. Got a little bit of burning, don't you? This is because when you are running at max performance, your muscles are needing more ATP than cellular respiration can provide. Is everybody clear on that? You simply cannot make enough ATP. Your heart has a limit on how fast it beats, right? You have a limit on how fast you can breathe. If you've really, really worked hard, really, really pushed it, you've really been out of breath, haven't you? Everybody here, I mean, just sprinting full speed for 100 meters, you pull up at the end of that, right? Because you need that oxygen. But since you're not getting enough of it, your muscles have switched over to the backup system. This is called fermentation. Fermentation does not require oxygen. Fermentation does not require oxygen. And that's a good thing, because it means you've got a backup. Now, there are different forms of fermentation. Humans use what's called lactic acid fermentation or lactate fermentation.
So, I don't know, it's hard to ask.
It's okay, just put it in your own
Like,
words.
I've heard of like lactic acid buildup.
Mhm.
What does that do?
Basically, the old hypothesis was, since you're really, really burning the calories, you're really, really going downtown, your muscles are switching over to the backup system more and more, and they're doing fermentation, right? Advantage, you don't need oxygen. The disadvantage, there's two. Number one, it is a lot less efficient. You cannot sustain life on fermentation. That big brain of ours just simply uses too much ATP. It has to have cellular respiration. The other side is the waste product of lactic acid fermentation is lactic acid. If you put a lot of lactic acid in the cytoplasm of your muscle cells, do you think that's going to do a lot of good? Probably not. The old idea was that the lactic acid buildup from this is what caused the burning and the soreness the next day. There's been more and more study on that, and it appears that it's not necessarily the lactic acid itself that's solely responsible. It plays a role. But you hear people saying hydrate and cleanse your muscles or flush your system or whatever new age gooey nonsense. What they're basically saying is you don't want to get this building up. You want to drink a lot of fluid so you stay hydrated and you flush it out. That help? Now this isn't the same type as fermentation as you use in brewing and bread. That's ethanol fermentation, right? You get alcohol and you get carbon dioxide. But you get the idea. So everybody okay with that? And you know what? We're almost done. That sound good? Next up, we've got carbohydrate storage. Carbohydrate storage. Now, when we talked about the macromolecules, we said that carbohydrates were beneficial because they're short-term energy, right? Remember that? They're the easiest food to break down, so that means it can happen quickly. If you're feeling a little run down, you want a quick burst of energy, you grab yourself something sugary and you're going to get it within a short period of time, right? It also means that when your blood sugar gets low, your body is going to need to tap a quick source to get it back up. That storage form is called glycogen. 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 Glycogen is essentially the animal version of starch. It basically branches. It's a long chain that branches. Starch in plants is typically a single chain that coils. Starch is fairly easy to break down, right? You actually start, believe it or not, digesting potato chips in your mouth. Did you know that? Spit, saliva, it actually has the enzyme amylase in it. As soon as you eat any kind of starchy food, it immediately starts to break it down. So you get the idea that starch is a storage form of carbohydrate, right? In animals, it's glycogen, primarily in your liver. So if you get low on blood sugar, a hormone called glucagon gets released. And glucagon is released by the pancreas, and it tells your liver, release some glucose. So the liver will very quickly break down the glycogen and get the glucose into the bloodstream. Everybody got that? So essentially, glycogen is the short-term energy storage. If you have an excess of energy, it's going to get stored as fat, lipids. When we start talking about connective tissue, and we talk about adipose tissue, you're going to see that there are essentially fat cells. Why does the body store energy in those two forms? Well, number one, glycogen is quick. When you need it fast, you need it now, you need glycogen. However, lipids store more energy per unit of mass than carbohydrates. In other words, if I burned a pound of lipids, I could get, say, twice as much energy as if I burned a pound of table sugar. You see the difference? Piper, do you get that? Okay. I couldn't tell if you're shivering, shaking, or not.
How does your body decide which one to burn?
What's the first one that it burns?
The sugar, the carbs.
Yeah, it's going to break down that glycogen first because it's fast. You still need energy. Your glycogen has been depleted. What's it going to switch to?
Your fat cells.
Yeah.
Is that why it's harder to burn fat?
Yep. The best weight loss treatment I have ever witnessed myself is, unfortunately, deathly illness. When I was in the hospital, I ended up losing a total of 54 pounds. Yeah. Because I was essentially had no appetite at all. They could bring in anything, and to me it tasted like warm, wet cardboard at best. I just didn't have a desire to eat. I was living off Ensure and my own body, essentially. Now remember, you also have proteins in food, right? But if you're not eating, your body breaks down the glycogen first, the lipids second, and what's left?
Your muscles.
Your muscles, proteins. It starts basically cannibalizing yourself. I got very fortunate in that I got out of the hospital before it got too bad. Y'all want to hear what cured me? You're going to laugh at this. I've been at home a day or two, and my wife goes in the kitchen to fix herself something. And for the first time in about 6 weeks, I actually wanted food. Specifically, I wanted what she was fixing. Because the wonderful, wonderful scent of frying bacon was coming from our kitchen. So she fried me 2 slices of bacon. And I wanted more. She ended up frying a whole pound of bacon. And she told me afterwards, she said, "That's when I knew you were going to be okay." And I said, "Well, you picked the right food." She said, "That's actually why I went out and bought it. I hoped it wouldn't get you hungry." So I owe my life to bacon. Okay. So y'all get the point, though. You consume proteins, lipids, and carbs. Carbs are going to be used by your body as the easy, quick energy source. And that's fine. They serve a role. Lipids, both breaking down and in your own body, are going to be the long term. Because they take a lot more to break down, don't they? But you're going to get more bang for your buck. So when you store it, you're going to store it in the adipose tissue, ADIPOSE, adipose tissue. That just means fat cells. And you got adipose tissue all around your body, but it does tend to accumulate in certain areas. Everybody understand how that goes? Do we get energy from proteins? Yes, but. And I say yes, but because your body doesn't necessarily want the energy from the proteins as much as it wants the amino acids. The amino acids are going to be the building blocks for your own proteins. For your own proteins. And there are a total of 20 amino acids. There's a total of 20 in life. And interestingly, your body only makes 11 of those. Now, your proteins require the other 9. Those 9 are called essential amino acids. You have to have them to survive. You have to have them to build protein. Where do you get them? Food. You have to eat a certain amount of protein to get those 9 essential amino acids. Can you get them if you eat a vegan diet? Yes, you can. You just have to be selective and make sure that what you are eating has the essential 9. Does everybody see that? It's easier to get protein from an animal source, but if you pick the right plants, you will be fine. So your body does get energy from breaking down those ingested proteins, doesn't it? But their primary use to the body is not as an energy source, and certainly not as energy storage. Make sure you've got that down in your notes somewhere. Make sure that you understand it in some way, shape, form, or fashion.
Can you repeat it, please?
I always wait until after I'm through with the whole thing and say, oh, just repeat that whole passage. Which part of the passage? So when you eat protein, when you eat sugar or carbs, it's short-term energy and short-term storage, right? Both in your body. When you eat lipids, it's long-term energy release and long-term energy storage in your body, right? When you eat proteins, you do get some energy, but the main utility of protein in the diet is for the non-essential amino acids, not as an energy source. Does your body use protein as a storage of energy? No. If you're starving, your body will self-cannibalize your protein, but it is not a normal standard form of energy storage. Is everybody clear on that? Did you have a question? Oh, yes.
If you eat like an excess of protein, where would your body store that?
You would break it down and you would consume it. Proteins have what we call an amino group, which is essentially ammonia. If you know anything about ammonia, you know it's toxic, right?
Yes.
I think we still got a couple of minutes. Let me check. We got 1 more minute, so hold on. You know where the ammonia goes? It's excreted in the form of urea, uric acid, which is in your urine. That help? Okay, that's it for chapter 4. We will meet in the lab at 11 o'clock.